What's up, FFO Penguins? So today we were doing a Unit 4 review session. It's again going to be our normal Q&A session. So we're going to do cell communication and the cell cycle. So I'm just trying to get everything all pulled up. Last time we had issues where y'all couldn't hear me. So please let me know if you all can't hear me so that I can make sure that I am doing everything on my part. I just tested it a moment ago and everything worked out okay. But you know how technology sometimes is. So if you can't hear me, please let me know. So we are going to do Unit 4 as I said before. So before we get in too deep, I want to remind you that there are a ton of resources on my website. So if you go to apbiopayments.weebly.com, I do, I've got the 396 page review guide. I've got, FRQ questions for every single FRQ that's from 2013 to current. I've got a daily review that I'm doing Monday through Friday on my Instagram. So every day I post questions every day, just try to help y'all to, make sure that we're getting everything we need. There are math that I'm doing every single day that I'm posting on TikTok as well as YouTube as well as Instagram. So anything you need is pretty much all there and it is free for you because my goal is that I just want to help students. So, let me know if you need anything. I'm always here for you. Okay. So starting tomorrow, we are 8 weeks away from the AP exam. Yes, there is exactly 8 weeks between tomorrow and the AP exam. So I do have an 8-week review guide to try to help you out. So as of today, March 9th, we are 57 days away from the exam. So this is kind of help give you a schedule. You can kind of pick and choose what you do on each of those days. But in terms of content review, if you're trying to use my stuff, you could use the CED TikToks, the unit review videos, or you can use like the topic questions that are in the review guide. To help you with your practice multiple choice questions, you've got the review guide unit multiple choice, practice FRQs. Those are all there. Now, as you get deeper into the later units, you can't really do them all. So I just said that you should be completing at least 4 FRQs on your review day, just so that you're working through the scoring guidelines and the simpler exemplar responses. And yes, we are 8 weeks until the exam. And then I've got my quizzes codes where I've got these are the full units of the quizzes to try to help you to review. Okay. And so here is that schedule. So you're welcome to screenshot if you need to, or you can always access this back on my website. It is posted for you. It's also pinned on my, oh, this one isn't pinned. But if you go to my website, you go to study plans. This is the 8-week study plan. So again, starting today, you would do that quizzes game to try to get you a gist of how you understand. And then you do like 3 days just topic reviews of either just overall topic review, or you could just do whatever your weaknesses are. And then you do multiple choice practice and you do free response practice. And then there was a day to break. And I follow that same schedule every week until the AP exam. So this week would be, this week would be Unit 1. Next week is Unit 2, then Unit 3. Then we go to Unit 4, Unit 5, 6, 7, and 8. And of course, here you see Cinco de Bio, the AP exam, of course. So today we're doing cell communication, cell cycle, and then there's a little bit of Q&A. There's not really any practice questions in this one, but if you go to my unit review, what I did last year, there are practice questions embedded where I go over the multiple choice as well as free response questions to try to help you out with the understanding of the information. And I'm so sorry about that background noise. My kids are playing downstairs. So the first step of our cell communication is reception. Reception has to do when our signaling molecule, our ligand, is going to bind to the receptor. So the receptor can be one of two different places. It can either be on the membrane, if we're looking at a water-soluble ligand, or it can be intracellular, so inside of the cytosol, if it's going to be like a testosterone or a steroid type hormone that's a lipid soluble hormone. So as soon as the signal or the ligand binds that receptor, it has a conformational shape change. So an example of a receptor would be a G protein coupled receptor. So as I said, steroid hormones are going to be released by simple diffusion. They'll just go strictly across that membrane. They do not need a transport protein because of the fact that they are lipid soluble and the plasma membrane is a lipid. The receptor is going to be intracellular, so it's going to be on the inside of the cell or inside of the nucleus. And then an example of that would be testosterone or estrogen. Those are just two different examples. On the exam, they can either just tell you, hey, there is a lipid soluble. They could tell you it's a steroid hormone. And then you would be responsible to know that that means that there's an intracellular receptor. You've watched all unit review videos, read a prep book. How do you make sure you leave all the info? We'll come back to those questions in just a sec. So protein hormone. These are going to be water soluble. So they're going to be released by exocytosis. So as we talked about before in Unit 2, you're going to have the protein synthesized by the rough ER. It's going to get packaged inside of a little transport vesicle that goes to the Golgi. The Golgi is then going to package and modify it. And then it leaves in a transport vesicle and moves to the plasma membrane. And then it's going to fuse plasma membrane, opening up. So there's that exocytosis taking place. The receptor for these are going to be extracellular. So they're going to be on that plasma membrane. And so the signaling molecule is going to bind to the receptor on the membrane. An example of this would be insulin. And if you don't know, insulin is going to lower your blood glucose level. So after you've consumed a meal, traditionally your body releases that insulin to help bring that glucose level down. It does it by signaling for your cells to put glucose transporters into the membrane, which then transports the glucose in so your body or your cells are able to go through cellular respiration to break down that glucose. So after we go through reception, after the signaling molecule has bound to the receptor and they go through that conformational shape change, we move over to transduction. So the goal of this is just to relay the message. The signaling molecule stays at the receptor. And so now we're seeing that the message is going to get relayed and it gets, one to the other, kind of like a cascade, to amplify the incoming signals and direct them to where they need to go. So there's one type that's called a phosphorylation cascade. This is going to use protein kinases. Protein kinases are going to phosphorylate. And so when you activate the protein kinase, it takes a phosphate off of an ATP molecule and it phosphorylates a protein. And then it's going to leave the ADP, and then that phosphorylation then causes the next one to be activated, which then causes the whole chain to get phosphorylated. So it's like if you walked down a hallway and you kept turning all the lights on, each of those molecules, those intermediates, would be phosphorylated. And so you've got this phosphate group on them. So now after that, you would need protein phosphatase in order to deactivate those and turn off all those lights. But so that's one example. Another we have is secondary messengers. So the primary messenger would be that signaling molecule, that ligand that bound to the receptor. That's your primary message. So the secondary messenger would be a small molecule that's able to move throughout the cytosol. So two examples would be, of course, calcium. So calcium ions, they're used in our muscle contraction. And so the calcium is stored inside of the sarcoplasmic reticulum in terms of your muscle, or it's just stored in the smooth ER. And then after there's a signal binds to it that causes the ion channel to open, that ion channel opens, which then releases the calcium out, and that calcium can move throughout the cytosol to go lead to other reactions, which is all part of that transduction. Another one we have is cyclic AMP. Cyclic AMP is just like ATP, except for it only has one phosphate instead of three phosphates, and it kind of has this circular cyclic kind of structure to it. And so it oftentimes will work in other signaling molecules. The AP exam, what they're going to end up doing is they're going to give you a pathway, you've never seen it before, and then you're going to apply this information to it. So they'll tell you there's a phosphorylation cascade, and you may have to understand what that means, or they'll tell you that a relay molecule is absent, and you'll have to understand what does that mean in order to figure out what went wrong in the system. Okay, and our last step is going to be response. So response has to do with what happens because of that signal and that transduction. What would be the final thing that happens in the cell? And so according to the CED, we've got cell growth, so the cell can grow. We have secretion of molecules, so it could cause there to be a release of some type of material from it. Maybe it's causing more insulin to be released. Maybe it's causing testosterone to be released. It's the secretion of something. Gene expression, it can turn on or turn off a gene. So we haven't gotten into gene expression yet, but they can act as transcription factors, and they can go into the nucleus and they can bind to the DNA to act as a transcription factor to turn on the transcription or the making of an mRNA from that segment of DNA. So then we can synthesize some sort of protein from it. So that can happen, or it can inhibit where it stops the transcription of a certain gene, so we don't make a certain protein. It can also that gene function or the phenotype, and then last but not least, apoptosis, which is Programmed cell death. So like the spaces between your fingers, the reason why you have those spaces, you originally had this big ball of cells, and your cells went through apoptosis, which allowed you to have these spaces between your fingers. It's also what happened to your tail that you had during embryonic development, and it's also what happens to frogs. That's how they lose their tails from tadpoles in metamorphosis. They go through apoptosis with that tail region. And so this was a diagram that was given on the 2012 exam, maybe. I can't remember. And so they wanted you to tell them all the different steps and then what was happening in those steps. So our first step, of course, is our reception. Because of the fact that this receptor is on the membrane, we know that this is a water-soluble protein. And so we would say that the ligand, or a protein hormone, is binding to that membrane protein, that membrane receptor, and that will then cause that receptor to change shape, which will initiate our transduction. So step 2 is that transduction. As you see, we've got these relay molecules. It's not that one thing is changing into another thing. It's that we are going to see the process where we're cascading to transduce the signal from one relay molecule to the next relay molecule, which you can see through shape changes of those relay molecules. Anytime you bind something to a protein, it always changes shape. And so that's something that I tell my students, and I assume you probably can know that also, that anytime you bind something to a protein, it will always change shape. That would be if we're looking at a transport protein, that's if we're looking at an enzyme, that's anytime you have a protein. If you bind something to it, it will always change shape, and that's important for us to know and recognize. And then notice here, we're talking about that we're now in the nucleus, and you see that there's this little molecule in our DNA. So this is telling me that the response is some type of transduction, where it's going, not transduction, sorry. It's going to act as a, oh my gosh, words, transcription factor, and it's going to initiate the transcription of those specific genes, okay? So our next part that we need to talk about is the cell cycle. So if you notice, I've got cells all over my shirt, which is actually a dress, but that's okay. And so you can see that they're going through these different phases. So we need to talk about the cell cycle and what makes up that cell cycle. So here you can see the overall diagram of the cell cycle. You have G1, which is our gap. You have that we've got the S phase, and then you see we have G2, which is our second gap, and then we of course have the M phase and cytokinesis. Okay, so interphase is going to be before mitosis, and cytokinesis is going to be after mitosis. There was an FRQ question in 2016 maybe, in which they asked you about the cell cycle, or actually they were asking about mitosis and what was similar between mitosis and meiosis. And so it was important that we understood that interphase was before and then cytokinesis was after these different processes. Okay? So in interphase, that G1 is going to be our gap 1. Okay, so this is going to be the cell growing, and it's going to be getting into that preparation to divide by allowing us to synthesize our organelles, synthesize the proteins, the RNA, as well as any of the building blocks. So it's just getting us ready. Now, it's important to notice that the cell does grow between all the phases. So although I specifically say that it's going to grow in G1 and G2, it actually grows through them all. But we do kind of help us remember that that G kind of for grow, you kind of think about it growing. S phase is going to be when we go through DNA synthesis, where we're going to synthesize a copy of that DNA. So we replicate the genetic material and all the centrosomes. And then the G2, again, we're cells growing, and we're synthesizing protein, the mRNA, and making the organelle, and is reorganizing its contents. It's hopefully going to be big enough now that we can go through this division stage, okay? So there are 4 steps to mitosis, or the mitotic phase, and this is going to be prophase, where we're going to be preparing to divide. So we're going to see that our chromatin that is just all unraveled, it's going to condense, and it's going to form these little chromosomes, okay? Now, because of the fact that we went through this S phase right here, they're now called sister chromatids. So this one and this one are both identical to each other. They have the exact same information because they are duplicates of each other. So our sister chromatids are going to find one another, and they're going to pair up at that centromere. So that's why that makes a little X shape, is because they're paired together at this little waist region on it called a centromere. And then our nuclear envelope is going to disintegrate. It'll go away, so we won't have that nuclear envelope anymore. And we're going to see that these spindle fibers are going to kind of move into their position on each side of each pole to prepare for that division. And these are little microtubules that are coming out of there. So what you see are these little microtubules coming out. So metaphase has to do with then these sister chromatids. They line up in the middle. So these little microtubules have attached to something called a kinetochore. It's a protein that surrounds that centromere. And so you see that it's kind of paired up to the middle, that centromere, if you may, of that sister chromatid. So it's going to have a tug of war. And as it does this tug of war, it's going to line these sister chromatids up in the middle. So someone says, what about prometaphase? Prometaphase is like kind of an intermediate phase between prophase and metaphase. It's not specifically required to be stated. Yes, but prometaphase would be right in this middle part when it's having that tug of war and you see these sister chromatids moving to their region. That's prometaphase, but you really don't have to describe it. The AP exam isn't even going to ask you what these phases are. They're going to ask you application questions about them. Anaphase is going to be our next step. Okay, so we've gotten the signal that everything's lined up in the middle, and there is a signal that causes the separase. It's going to allow for the two sister chromatids to separate. It breaks that cohesin molecule that's holding them together, and it's going to allow them to separate. So the microtubules are going to go through deep polymerizing. So they're this big polymer, and they're going to break down, and they break down, they believe, from each of the ends. Okay, and so as they break down, it's kind of like a fishing pole, and it's going to reel these two chromosomes. Now they're called chromosomes because they're apart from each other. They're going to reel the two chromosomes apart and bring them to opposite poles. And so that's where we see our sister chromatids are going to be pulled apart to opposite poles. And then once they're in there, we make our two new nuclei. So in telophase, or some people call it telophase. I say telophase. You're going to see that we're going to have this new membrane forming around those. So that is the end of mitosis. Notice this cell is still a one unit. So your cells can go through mitosis without going through cytokinesis. And so that happens with muscle cells. That's why they're multinucleated, because they have this mitosis that takes place without their cytokinesis. And the cytokinesis is going to be the last part where we're going to actually see the cytoplasm separating. So we divide the cytoplasm, and that forms our two new cells. And now this was kind of showing you an animal cell. A plant cell, slightly different, in which the cytokinesis of it is going to be formed by, they're going to make a cell plate, and then that cell plate forms the cell wall that then would separate the two cells. But in animal cells, there's an actin in there. There's actin that is a microfilament, and it just kind of basically squeezes tighter and tighter and tighter and tighter until finally we have two new cells that kind of pop apart from each other. So during this process, there's going to be these different checkpoints, these different things that stop to make sure that everything is rolling like it should roll, that we should keep on going, because if there is an error, we need to stop the process before something bad happens to our cells. So my textbook always talks about a washing machine and how your washing machine has all these little internal and external signals. And those signals kind of determine, hey, should I move on? So for example, you put the clothes into your washing machine and you close the door and you turn it on, right? The water starts filling into your washing machine and it fills all the way up. How does it know when to stop filling? It's either A, that there's a certain amount of water put in there and it keeps track of that, or B, it has to do with the weight, or C, there's probably a sensor in there. And when the water gets to that sensor level, it probably says, oh, we have enough water, turn off. So the same type of thing is going to have to happen within your cell to say, okay, we should move into this next phase, or wait a minute, don't move into the next phase. It's not ready yet. We don't have enough materials. We don't have enough resources, whatever the situation is in that checkpoint. So our first checkpoint is the G1 checkpoint. This one determines should the cell divide. So it'll either has a growth factor that is bound. So a growth factor is a signaling molecule that binds that says, hey, I need the cell to divide. And is also making sure, does it have enough resources to divide? If there's not enough resources, it really shouldn't be dividing. And it's going to check to make sure there's no DNA damage. If it can't get past that that G1 phase, it exits into something called G0 or G naught. Okay, this is a non-dividing state. Most of your cells just kind of chill out in this non-dividing state if they're not dividing. They're just hanging out. Okay, your liver cell is always in that stage. Your nervous cells, as well as your muscle cells, these are forever. Once you've done growing, these kind of stay in that phase. And the liver can be caused to switch back out if there's external signals. Okay, so here you can see it. We're going to see that there is a certain type of cyclin in there that's going to bind, and if it's activated, then we're going to move past the G1 and we'll go into that S phase if it has the phosphorylation of these certain genes. Okay, now again, we need to have that signal bind to our cyclin-dependent kinase to allow us to move into that phase. Okay. So our G2 checkpoint is making sure that we have replicated all the DNA. So if you remember, we go G1, S, and then G2. So our G2 checkpoint is making sure that was there any damage made to the DNA while it replicated? Did it completely replicate? Because I need there to be 2 copies of every single piece of DNA so that when I go through mitosis, each cell will have an identical copy of the DNA. So I need there to be that step that says, hey, DNA is good. Hey, I've replicated all that DNA. If it detects any problems, it's going to hold the cell cycle until we can either repair it. Usually P53 will repair it, and making sure that we're done with the replication. So here you can kind of see that there's this damage. P53 is going to come in. It inhibits CDK. And so since there's a CDK inhibitor, it stops that process from moving on. So it kind of pauses the phase so it doesn't move into the next step. And then you have the M checkpoint. The M checkpoint is meant to make sure that every sister chromatid is attached to a kinetochore microtubule. If I do not have a kinetochore microtubule to both sides, when it pulls, it's going to pull both of them to one side. Versus if we have one attached to both sides, it's going to move equally to either side. Okay, we need to make sure that there is a sister chromatid attached to every microtubule so we have equal cell division during mitosis. So here you can kind of see that we have our gene that's attached, making sure the cohesin is together. If we do have all of them attached, that's then going to cause this little proteasome to break it down so we can then use separase to separate the cohesin molecule so that the two can move to opposite poles. Okay, so that is all that's in unit 4. Like unit 4 is a very short unit. It's just cell communication, and then it has to do with mitosis. That's all that's in 4. And there was only one question submitted online. So you've got time that if you want to type your question into the chat, you're welcome to type those into the chat, and I'll answer those as we go. I'm first going to answer the one that was submitted online, and it says, can we talk about cancer and cell disruption? Okay, so in terms of our cancer, okay, oftentimes cancer comes from mutation. There's some type of mutation that takes place, and because of that mutation, the cell replicates and keeps that mutation in there. And so sometimes we see it as a proto-oncogene. So basically all of our cells, they're either proto-oncogenes, or they're going to be cell, what's the word I'm looking for? Proto-oncogenes that become oncogenes, or it's going to be a tumor suppressor gene. So our proto-oncogenes are normal cells for cell division. Okay, their job is to allow the cell to make the proteins to go through cell division. If we have a mutation in one of those genes, that can cause my proto-oncogene to become an oncogene. So like the RAS protein. And so what it does is it makes extra copies of the signal that says, hey, time to divide. Hey, time to divide, even though the cell shouldn't be dividing. And so that's where we see the proto-oncogene becoming an oncogene. The other type of example we have is a tumor suppressor protein. Tumor suppressor protein is kind of like P53, okay? If we have a mutation in our DNA, right, we don't want the cell to divide with that error. So our tumor suppressor protein, which is like P53, is going to halt the cell cycle. If there's a mutation in P53, it's going to allow this cell to divide with this error. And so that's another way we can get cancer is due to the fact that P53 has a mutation, and that leads to cells dividing with DNA damage, which then leads to that cancerous problem. Okay, so you can see we have the initial mutation and activates a negative cell cycle regulator. So that's our pro-oncogene becoming oncogene. It overactivates a positive cell cycle regulator. Again, that's an oncogene. Here we can see a genome stability factors. That would probably be our tumor suppressor protein. And then as these mutations accumulate, that leads us into some type of cancerous cell. Okay. So here you can kind of see different things about it. So like a cancer cell, normal cells, you have to give them growth factor in order for them to divide. If you don't have growth factor, then they won't divide. And they also have like adhesion where the cells have to be bound to some type of substrate. So traditionally, if you were to do a cell project, you wanted to grow cells, they're growing at the bottom of these little well plates. And you have to put in an enzyme in there. I think it's like trypsin or something. And that releases the cells from the bottom of your cell plate, so you'll be able to grow them. Versus cancerous cells, they don't have to be bound to anything. They can be free floating in a solution. If you've read the Immortal Life of Henrietta Lacks, you remember they talked about that it was replicating while in the solution because those were cancerous cells. Also, they don't need a growth factor. If they have growth factor, if they don't have a growth factor, they're still going to continuously divide. And so that's where we see with our cancerous cells and cell communication. So somebody says, you've watched all the videos, all the AP classrooms, you've read a prep work. How do you make sure you have all the info needed for the exam? Um, so it's all about practicing in multiple different ways. So the more that you interact with the information, the more solidified your understanding is going to be. On the AP exam, they're still going to give you things you don't know. It's going to be from some article, and you're going to need to just apply the biology to it. So do not be freaked out when you read something that you don't know. Just kind of lean back, take a second, breathe, and think about the stuff that you do know. Find something in that question that you do know. I don't really think there's a way that you can know everything there's possibly going to be on this exam. It's all about applying the things that you do know. And if you've watched all the videos, you've watched all the AP Classroom videos, and you've read a practice book, I think you just need to do a little bit more like active studying. So going through practice questions, finding where your errors are in those practice questions, and kind of working through those mistakes and those misunderstandings, seeing is it an application issue or is it a content issue, and just kind of keep working through it. You do practice multiple choice questions and FRQs. Check the understanding. There's a bunch on the Crack AP website. Yep, so you can go through the Crack AP website. Somebody says cyclins and cyclin-dependent kinases. So cyclins are synthesized throughout the process of the cell cycle. So you have this increase of the cyclin molecules. And as those cyclin molecules increase, once they reach a certain level, that's when allows them to bind to cyclin-dependent kinase. And the kinase, as we said before with protein kinase, their job is phosphorylation. So the cyclin-dependent kinases are going to phosphorylate, and that allows them to move from one phase of the cell cycle to another phase of the cell cycle. So they're going to kind of keep moving from one phase to the next. There's multiple different CDKs. There's multiple different cyclins. They're going to give you application questions with those, and it's important that you understand that a CDK just means that it's binding to the cyclin, and it's requiring that cyclin to be there in order for it to do its job, and they'll tell you in the prompt what that CDK's job is. What's the biggest difference between G1 and G2? Like how you can tell the difference. So the biggest difference is going to be the amount of DNA. So G1 is going to have the normal level. So if they tell you that there's 46 chromatids, then you would have 46 chromatids in G1, but you would have 92 chromatids in G2 because G2 is going to be after the S phase. So it's after you've gone through that cell division and you've replicated the DNA. And so that would be the biggest difference you would see between those two. Cells still growing, is still preparing. That's the gist of what you really need to focus on, is just that we do have to go through that long process of growing and dividing. Growing in order for us to get into that divide stage. Will the cell only be sent to G0 after G1? Can it go to G0 at any point during interphase? I believe the only point that it goes to G0 is if it doesn't get the go ahead at the G1 checkpoint. So the G1 checkpoint is more or less the give or take. So everything I've ever read about G1 states that if you don't get past G1, you probably won't divide. So I assume that if you don't get past G1, you'll just enter into that G0 or that non-dividing state. You're welcome, Crystal. Question about the month of April. What should teachers be doing? My teacher is just about done with Unit 7. So your teacher probably has a plan. If you're almost done with Unit 7 at this point, that means that you're probably going to get through Unit 8 in March, which means you'll probably spend April reviewing. So they're either going to give you some type of review project that you work on, which is what my students are doing. They have a review project. I play review games with them each day. We do FRQ, we do tips and tricks. I do a mock exam in class. So talk to your teacher, say, hey, are you going to plan on doing a mock exam for us in class so that we don't have to come on like a Saturday? My students have the option. They can either come on a Saturday, and then they also will, every single one of them will take one during class. I think it's about a week before the AP exam. I'm going to have them take it so that I can give them that last minute feedback of, okay, these are your strengths. These are your weaknesses. This is what you need to focus on the next week so that you are as prepared as possible for the exam. The more that you can practice, the best. I also do something called Race to the Slopes with my students, and each day we do practice questions. So every Tuesday and Thursday, they're right now, they started back in February. They take a 9-question review quiz, and it's over any topic throughout the entire curriculum, just random, honestly. And they are competing to see who can get the most questions right. That's why I call it a race to the slopes. Tips for students who want to start reviewing. So as I said at the very beginning, I've got this little review plan put up for you. So you can review in whatever method kind of works best for you. My personal opinion is that you need to do something that's going to help you to review all the information. Okay, so let's say like here with this example. Let's say we do some little quick quiz to kind of see if we understand something. You can rotate this around. but you do something to review the information, whether it's you read through that section of your textbook, you watch a video, you read a section in the review book, whatever it is that you need to just get a holistic review of the content. Okay. And then you want to fine tune it on the things that you don't really understand fully. So I really struggle with, I don't know, cell communication. So you would then sit there and you would do a bunch of cell communication videos or resources in your textbook, or you would do some practice questions. You would do stuff that kind of helps hone in on that misunderstanding you have or that weakness. And then you would do like multiple choice and FRQ holistically. So that's kind of the way that I've set them up. It's all on you and what time you're looking at this. If you're watching this on March 9th, logically you could be doing 1 unit a week. If you don't see this until like, I don't know, the first week of April, then you're probably going to have to do like 2 units a week to kind of get focused. And if you're seeing this 10 days before the AP exam, you're probably going to be needing to do a little bit more of just fine tuning or overall reviewing. So it's depending on you and how much time you're giving yourself to review. Same thing is if you're thinking about reviewing, like how long are you spending each day? Are you spending like 10 minutes reviewing? Because if you're spending 10 minutes, you want to start earlier. If you're planning on spending like hours studying, then yeah, you could start later, but you're not going to retain as much information by starting later as you would by starting earlier. Okay. You're welcome, Alec. How behind is my class if we just finished unit 4 and you still have 5 and 6 to do? So you could probably get 5 done in March, and then 6 could be done in April. So you're probably going to be on track to be prepared for the exam. You probably want to do a little bit of self-studying on your own, just because of the fact that you may not have time in class to review. But I would just be each night just doing a little bit of review, just kind of cycling back, making sure that you've understood the stuff that you've looked at before. If you've prepared all school year, then you're really not going to need to be doing a lot of hardcore review in this time. You're probably going to do a little bit more of just kind of quick review, because of the fact that you've been preparing all year. You don't really need to relearn the information. And then clearly I've just lost my PowerPoint. You don't really need to be relearning all the information because of the fact that you've already learned it all, right? This thing is like all way behind. Sorry. You've learned everything already. So like, it's just a matter of refreshing and bringing it back to the surface. I was trying to get that Q&A slide, but I think I went too far. I did go too far. There it is. Okay. So we got to the end. Okay. So what other questions do we have? What are G protein coupled receptors? So G protein coupled receptors are a type of receptor that you're going to find on the plasma membrane. Fun fact, they have 7 passes of the membrane, so they kind of go back and forth 7 times. And so a G protein coupled receptor traditionally works with a G protein that works with a GTP molecule. So ATP is adenosine triphosphate and GTP is guanosine triphosphate. So it's just a difference in the nitrogenous base. But it's otherwise still the same. And so the GTP molecule is going to activate a G protein that will get activated due to the G protein coupled receptor. Traditionally, that G protein then works with an adenylyl cyclase, and the adenylyl cyclase is going to convert the ATP into cyclic AMP. And that is our secondary messenger that we mentioned before. So that whole process from that G protein coupled receptor all the way through that making of the cyclic AMP, that's all that transduction part. And so it's just looking at that, which actually cyclic AMP is still part of transduction. Okay, if your class started with unit 7 and 8 and then you went in order, should I start studying with unit 7? It's up to you. 7 is kind of the stuff you don't remember as much, but as long as you hit that unit 7 before you take the AP exam, it's really not going to be a problem. So you could probably do unit 1 through 8 if you wanted to, but you could also start with unit 7. It all depends on you and how you feel. Do you think the AP reviews in College Board are helpful for prep? Well, those were written by people that are working for College Board. So theoretically, that is the information that College Board wants to make sure that you know. Also, me and any of the other creators out there, we're looking at the CED, the same CED that you are able to look at. Literally Google AP Bio CED, and you're going to find the curriculum that all the creators are using, all the teachers are using, everyone is using to try to prepare you for the exam. So College Board theoretically has a more inside track of what they want you to know. So theoretically, those videos would be more helpful, but some people don't find them to be the most entertaining to watch. You know, like a crash course video is like so entertaining because Hank Green is just all over the place. He's got all these little animations. So it's just depending on you and your learning style and who makes the most sense to you. Um, Khan Academy, is it a good source for reviewing and studying? That is much to be argued. Most AP Bio teachers in the world do not agree with Khan Academy, but it's a free resource. So who am I to argue with free resources? So if you want to use Khan Academy, go for it. Um, Anytime that you're able to get extra help to review and learn information, that's really not a problem. The issue is that they sometimes have wrong information in their stuff, and then they also still hit stuff that isn't in the curriculum. So your class is currently Unit 6. You're finding is most difficult. The teacher made a self-learn 8. Do you think Unit 8 is self-study? Yeah, Unit 8 is an honest, really good self-study. It's ecology. You went through middle school. You know predator-prey relationships. If I said the word euparasite, you probably know what a parasite is. You probably know trophic structures. You know your primary producers, your secondary, like your primary consumers. You know food webs, food chains. For the most part, that's not going to be new information for you. And it's kind of logical information versus like Unit 6 is a very difficult unit because you've got DNA, RNA, these enzymes that go do these different things. So I think that Unit 8 is pretty easy to do a self-study. What units show up most on the AP exam? The units that show up most on the AP exam is Unit 3, 6, and 7. If you look at the CED, it actually has a chart for you, which I don't have it played out right now, but they have a chart on it, and on that chart, it tells you the exact percentages that are in each of the units. So here is that CED. I don't know if I can get to the part on the CED fast enough, if it's going to cooperate for me. Um, but in there, you can actually scroll down and you can see the amount of each of the units that are on the exam. So here we go. So let me zoom that up so it's nice and big for you. So you can see that Unit 3 has 12 to 16%. Unit 6 is 12 to 16%, and then natural selection is 13 to 20. So those are kind of like the bigger ones. Like 15 isn't that far apart. So like it's pretty evenly distributed between all the units. But if you're trying to think like what is the one that shows up the most, technically it's like 3, 6, and 7 is the one that shows up the most. What other questions do we have? Do you have any thoughts on the Campbell textbook? You struggle with staying focused on reading long sections. So I personally love the Campbell bio book. That's like my favorite book to read and to use for resources. So with that, you're going to see that we have you have trouble reading long sections at once. What I find is that if you read a section and then you summarize it, that kind of helps out. You're also going to find that it's easier if you kind of jot notes down as you go and kind of have something to keep you focused on it. It all depends on you. Like it's all kind of what works best for you. My students have a reading guide that they can choose to use, so maybe your teacher has a reading guide that they can use to help you. Yeah. Okay, so last year's exam you've heard was majority on ecology. You think it's like that again? I don't think it will be like that again. They do honestly follow the system. My kids are being so loud. I'm sorry, y'all. So usually we do have, or they follow that percent, and they're going to follow that percent to a T about the number of sections there's going to be in each unit. They don't really deviate from that. Now, you may feel that it's going to be very ecology based because like stuff may be seated in ecology and they may be giving an ecological example, and then they ask you a cell communication question, or they may ask you a cell energetic question. So don't get fooled by saying, oh, well, this is ecology because it has to do with organisms. Well, you know, we're looking at biology. The entire year is on organisms. Is there a way to predict what unit the FRQs will be like? Yeah, so if you go to the end of the CED, you can actually see what the FRQs are going to be like. I don't know if I can get there fast enough. I may have just passed it. No, science practices. But yeah, there is a section of this that tells you exactly how the FRQs are going to be laid out. So here you see that the first FRQ is going to be interpreting and evaluating experimental results. You know that the first part is going to be describing and explaining your concepts. So this will be about the biology. B is going to be about experimental design. C is going to be analyzing some type of data. D is making predictions. So like you can see clearly that, what one's going to look like. 2 is going to be the same idea, except for now you're constructing a graph. 3 is going to be giving you an experiment, and you're then going to be applying that experimental procedures to it, as well as predicting some information. 4 is conceptual, so they're going to have you talk about changes in a certain system, and then, of course, describing and explaining that system. 5 is going to be some type of relationship, and you'll do some type of drawing with it. 1 year you had to draw what a certain field was going to look like in terms of the organisms. You've had to add an invasive species to a food web. You've had to draw an X on a phylogenetic tree. You've had to make a cladogram before. So there's a lot of different things that you might have to do for part 5. And then 6 is going to just be analyzing data. So there's an experiment and you're analyzing the data to it. So they do clearly state out how your FRQs are going to be. And if you want practice on the FRQs, AP Central has all the FRQs that they've ever released back to like 1999. And you can use those. They've got the question, they've got the scoring guideline, they have exemplar responses. So you can see what a kid who got a perfect score on it got. You can also go to the chief reader notes and you can see what students across the nation struggle with in it. So it really does lay out exactly what, you know, you can do to help. And if you're wanting me, I have FRQ Fridays where I've gone through every FRQ and I've explained to you where they get their points and what I would have done going upon it. So it doesn't look like we have any new questions that have popped into the chat. So if you have any other questions, please type those into the chat. I'm going to show you my website real fast just so that you see it in case like you needed to see it while I'm waiting for people to post some last minute questions into the chat. But if you have no more questions, once I'm done doing this, I'm probably just going to log out of it just because of the fact that, you know, if you don't need me, then I'm not going to keep saying live for y'all. I'm trying to get back to the YouTube. I'm sorry, so I can see your messages. Okay. So here you can see the Instagram. Last year's crew said that these were their most helpful resources of mine. So the unit review videos, the TikTok videos, the quizzes games, these are all posted here for you. Then we've got the review videos are also linked here. The TikTok videos, the review guide, that 396-page review guide that I posted, or I told you about at the beginning, it's right here. You click there, and I've got I-can statements. I've got all of the units broken up by topic questions. There's unit reviews in there. There's FRQ questions, multiple choice questions. Everything is clear cut in there for you. There's review games. There's the Insta-Review that I'm posting every day on Instagram. Somebody just wrote in the chat, where do I post my FRQ Fridays? The FRQ Fridays were made last year. I haven't made the new ones for this year. So I was planning on maybe starting this upcoming Friday and doing one each week so that you have those being posted. But they will be posted here. So if you click on this, it'll take you to where you either look at it by unit or you can look at it by their year. So there's all easy organized for you. There's the review PowerPoints. Pure Penguins, these are things that are made by other students that they said, hey, yeah, you can put this on your website to help other students. This is the 10-day crunch. Basically, I just put each of the review videos in there, and it's just meant as their crunch session. I've got the one-pagers. There's all the math videos that I did last year where I worked through every math question from the curriculum, the CD. And I've done an example of every formula sheet on there. And then there's the results from last year's students. So if you're wanting to be like nosy, say, hey, you know, a student who got a 5 last year, which resources did they use? You can access that there. You can click and it gives you the whole the whole Excel file that has all 1,000 students who answered, like asked me questions, all the 1,000 students who answered it, telling me what they did to prepare for the exam. And so I'm asking them more questions this year, or asking you more questions this year if you do that for them. So, and then if you're wanting exam calculator, I did make an exam calculator to kind of help you to use, you know, if you did a practice exam, what, you know, generally your exam would be. So all that is available as you see there. And look, 56 days and 16 hours till the AP exam. Oh my. All right, so let's see what questions have come in the chat. So somebody asked about the FRQ prize. I already answered that. Where did you say College Board released the FRQs again? So if you go to Google and you type in, actually, let me pull over my Google. I really shouldn't be doing this live, but that's okay. So if you just type in AP Bio FRQ, you will find all the exam questions right here. So they just have them on their website. And so here's, you know, 2024. That's where you find the questions. The scoring guidelines are there. The Chief Reader Report, where she explains everything that was, helpful from last year, like what students did well, what they didn't do well. You can see how the nation did on those FRQs last year. You can see how many students got a 3, 4, 5 on the exam. And then they have the exemplar responses. So there's usually a student who got a perfect score in there, and they tell you why the student got the perfect score. You also have usually like a mid-range person, so someone who got like, I don't know, 8 or 7 FRQ points, and there's usually somebody who got like 3 FRQ points. You can kind of see the different levels to see what the student did, well, or what they might have misunderstood that caused them to not get points. So those are all there for you, and they have them going back to like 1999. So there's tons of those questions up there for you. Okay, what other questions do we see? For units 7 and 8, do you think there should be a lot of heavy studying like the other units? That's up to you. If you are actively preparing for your class, then you probably don't need to have as much hardcore studying, because the fact that you already studied and it should be relatively recent. I would be looking over the information, but you may not need to do as much hardcore studying because it would be recent for you. Somebody earlier said that 7 and 8 was their first 2 units that they studied. So they probably would have to do a little bit more hardcore because they, learned those a long time ago. Hi, what is a good resource you can find multiple choice questions? AP Classroom. Honestly, AP Classroom is going to be the only place you can go to get questions that are similar to the exam. And I'm so sorry about my kids. Usually I make them go somewhere, but today I guess they're playing in the living room. Okay, so what other questions do we have? I can see that there is a big lag on my screen. So I assume that there's also that same big lag on your screen. Oh, wait, maybe not. How far are we advanced? I don't know. Anyway, so I hope that today was helpful. If you need anything else, you know where to find me. Every day I do have a little question box on my Instagram, so you can post your questions into that Instagram, so you can ask them. But since there's no more questions going into the chat, I'm going to go with that you all are good, that you don't need me. So I hope that this was helpful. And remember